scientists have developed GLM, an AI system that decodes the complex language of genomics from extensive microbial data. This innovation enables a deeper understanding of gene functions and regulations, leading to new discoveries in genomics. GLM exemplifies the potential of AI in advancing life sciences and tackling global challenges. Artificial intelligence AI systems like ChatGTP have taken the world by storm. There isn't much they don't have a hand in, from recommending the next binge-worthy TV show to helping navigate through traffic. But can AI systems learn the language of life and help biologists reveal exciting breakthroughs in science? In a new study published in Nature Communications, an interdisciplinary team of researchers in the Department of Organismic and Evolutionary Biology (OEB) at Harvard have pioneered an artificial intelligence AI system capable of deciphering the intricate language of genomics. Genomic language is the source code of biology. It describes the biological functions and regulatory grammar encoded in genomes. The researchers asked, can we develop an AI engine to read the genomic language and become fluent in the language, understanding the meaning or functions and regulations of genes? The team fed the microbial metagenomic dataset, the largest and most diverse genomic dataset available, to the machine to create the Genomic Language Model (GLM). In biology, we have a dictionary of known words, and researchers work within those known words. The problem is that this fraction of known words constitutes less than 1% of biological sequences. The quantity and diversity of genomic data is exploding, but humans are incapable of processing such a large amount of complex data. Large language models (LLMs) like GPT-4 learn meanings of words by processing massive amounts of diverse text data that enables understanding the relationships between words. Genomic Language Model (GLM) learns from highly diverse metagenomic data sourced from microbes inhabiting various environments, including the ocean, soil, and human gut. With this data, GLM learns to understand the functional semantics and regulatory syntax of each gene by learning the relationship between the gene and its genomic context. GLM, like LLMs, is a self-supervised model. This means that it learns meaningful representations of genes from data alone and does not require human-assigned labels. Researchers have sequenced some of the most commonly studied organisms like people, E. coli, and fruit flies. However, even for the most studied genomes, the majority of the genes remain poorly characterized. Researchers learned so much in this revolutionary age of omics, including how much we don't know. How can we glean meaning from something without relying on a proverbial dictionary? How do we better understand the content and context of a genome? The study demonstrates that GLM learns enzymatic functions and co-regulated gene modules called operands and provides genomic context that can predict gene function. The model also learns taxonomic information and context dependencies of gene functions. Strikingly, GLM does not know which enzyme it is seeing, nor what bacteria the sequence comes from. However, because it has seen many sequences and understands the evolutionary relationships between the sequences during training, it is able to derive the functional and evolutionary relationships between sequences. Like words, genes can have different meanings depending on the context they are found in. Conversely, highly differentiated genes can be synonymous in function. GLM allows for a much more nuanced framework for understanding gene function. This is in contrast to the existing method of one-to-one mapping from sequence to annotation, which is not representative of the dynamic and context-dependent nature of the genomic language. In the lab, researchers are stuck in a step-by-step process of finding a gene, making a protein, purifying it, characterizing it, etc. And so we kind of discover only what we already know. GLM, however, allows biologists to look at the context of an unknown gene and its role when it's often found in similar groups of genes. The model can tell researchers that these groups of genes work together to achieve something, and it can provide the answers that do not appear in the dictionary. Genomic context contains critical information for understanding the evolutionary history and evolutionary trajectories of different proteins and genes. Ultimately, GLM learns this contextual information to help researchers understand the functions of genes that previously were unannotated. Traditional functional annotation methods typically focus on one protein at a time, ignoring the interactions across proteins. GLM represents a major advancement by integrating the concept of gene neighborhoods with language models, thereby providing a more comprehensive view of protein interactions. With genomic language modeling, biologists can discover new genomic patterns and uncover novel biology. GLM is a significant milestone in interdisciplinary collaboration, driving advancements in the life sciences. With GLM, we can gain new insights into poorly annotated genomes. GLM can also guide experimental validation of functions and enable discoveries of novel functions and biological mechanisms. We hope GLM can accelerate the discovery of novel biotechnological solutions for climate change and bioeconomy. Thank you for watching. If you've enjoyed our videos, subscribe to our channel for there's more to come.